Imagine you were trying to understand the layout, the culture, the whole economy of a massive city, like New York or Tokyo.
Okay, I'm picturing it.
But instead of, walking the streets or looking at a satellite map, you take the entire city, throw it into a giant cosmic blender, and then study the resulting sludge.
Wow. That is a very messy image.
Right. And you'd analyze this sludge and find out the city is like 20% concrete, 10% glass, and, I don't know, a fraction of a percent pizza dough.
Yeah, you have all the raw ingredients.
Exactly. But you have completely lost the architecture. You don't know where the financial district is, where the parks are, or how any of these neighborhoods actually interact with each other.
And as crazy as that sounds, for a really long time, this blender approach is essentially how the medical field has studied cancer genetics.
Which is wild to think about.
It is. Doctors take a piece of a tumor, grind it up, and sequence the RNA of the whole mixture. It's a process called bulk RNA sequencing. And while it does tell us what ingredients are in the tumor, it destroys all the context of how those cells were actually arranged in the human body.
And today, our mission for this deep dive is to explore a groundbreaking 2024 study published in Nature Communications that finally throws away the blender.
Long overdue, honestly.
Seriously.
Yeah.
The researchers used this revolutionary technology called spatial transcriptomics to map out one of the most notoriously difficult cancers out there, triple-negative breast cancer, or TNBC.
Right.
They actually managed to keep the cellular city entirely intact to see exactly which cells live where. And, spoiler alert, it is completely rewriting how we understand the disease.
Which is a huge, vital breakthrough. I mean, considering TNBC accounts for 15 to 20% of all breast cancers, and it traditionally carries a much poorer prognosis than other types.
Right, and the challenge with TNBC is baked right into its name, isn't it?
Exactly. Triple negative is actually a diagnosis of exclusion. It just means the cancer cells are missing 3 specific receptors.
So they don't have estrogen receptors.
Right. They don't have progesterone receptors, and they don't overproduce a protein called HER2.
So if we think of those 3 receptors as locks on the surface of a cancer cell, doctors have developed highly effective keys to target them over the years, like hormone therapies that lock in and shut the cancer down.
Which is great if you have the locks, but because TNBC lacks all 3 locks, those standard keys just don't work.
It's like trying to use a remote control on a television that doesn't even have an infrared sensor.
That's a perfect analogy, and because it's defined entirely by what it's not, TNBC isn't just one uniform disease.
It's a catch-all.
Totally. It's an incredibly heterogeneous umbrella category for several very different types of tumors.
But the old blender method, the bulk sequencing, did manage to give us a rough breakdown of this chaos, right?
It did, yeah. It revealed 5 distinct molecular subtypes inside that umbrella.
Okay, let me list these out for you listening. We have the basal-like, immunomodulatory, luminal androgen receptor, mesenchymal, and mesenchymal stem-like subtypes.
That's a mouthful.
It really is. But even knowing those 5 distinct flavors of the smoothie, we were still flying blind geographically.
Exactly. We knew who was in the city, but not who was living next door to whom.
So the researchers behind this study took samples from 94 early-stage TNBC patients and analyzed them using this spatial transcriptomics. Let's unpack the actual mechanics of this map. How does this technology actually keep the city intact?
The physical process is honestly fascinating. They took fresh frozen surgical samples from these 94 patients and they sliced the tissue incredibly thin.
Like microscopic then?
Exactly. And then they placed these fragile tissue slices onto special glass microarrays. You can think of these arrays as like high-resolution satellite grids.
Okay.
The glass is printed with thousands of microscopic spots, and each of those spots contains millions of unique genetic barcodes.
So when the tissue sits on the slide, the RNA from the cells in that exact physical location gets permanently stamped with a specific barcode.
You've got it. So when the computers sequence it later, they can trace every single genetic signal back to its exact microscopic zip code on the glass.
That is incredible. And the scale of this mapping is staggering. Across the cohort, they analyzed 270,310 of these barcoded spots.
Yeah, the data volume is massive, and each individual spot is about 100 micrometers in diameter.
Which is roughly the thickness of a single human hair, right?
Yep, exactly.
Wait, I'm trying to picture the math on that. 100 micrometers is definitely tiny. But a single human cell is much, much smaller than a hair.
Right.
The study actually notes that each of these spots can capture the RNA of up to 200 individual cells at once.
That's true.
So if there are 200 cells mashed together in one spot on the grid, Aren't we just making 270,000 tiny mini smoothies? How does making the blender smaller actually solve the fundamental problem of unmixing the cells?
That is the pivotal hurdle of the entire technology. You are entirely correct that each spot is a tiny mixture of cells.
So how do they fix it?
The solution comes from a layer of computational magic called spatial deconvolution.
Okay, break down how that math works for us.
Let's use a visual analogy. Imagine you have a tiny cup of mixed paint, let's say a murky purple.
Okay, I see it.
Theoretically, that purple is made of red and blue paint. But to figure out the exact ratio in this specific cup, the researchers brought in expert pathologists.
Oh, to look at the actual cells.
Right. They looked at traditional physically stained slides of these exact same tumor tissues. The classic pink and purple slides you see in biology textbooks.
The visual map of the physical structure.
Yes. The pathologists manually annotated the distinct physical geography. They'd draw a circle around pure tumor cells, highlight the connective tissue, point out the blood vessels.
I see.
And then the researchers fed both sets of data into an algorithm. By cross-referencing the jumbled genetic data from our mini smoothies with the incredibly precise visual map drawn by the pathologists, the algorithms learned to mathematically unmix the purple paint.
Oh, wow. So they train a mathematical model to recognize what the RNA of a pure tumor looks like versus the RNA of a pure blood vessel.
Exactly. So for any given 100 micrometer spot, the computer can look at the mixture and confidently say, this specific spot is 60% tumor, 30% supportive tissue, and 10% immune cells.
That's brilliant. So the algorithm successfully deconvoluted the spots into up to 9 specific microscopic neighborhoods.
Spot by painstaking spot across 94 different patients. They could map the tumor cells, the fat tissue, the vessels, the dead tissue, the immune cells, everything.
So just to step back and recap what we've unpacked so far for you listening, we have completely abandoned the blender approach.
Thanks.
By using glass slides stamped with microscopic barcodes and powerful math that unmixes the genetic signals, we now have a high-definition map of the entire tumor ecosystem.
A literal map.
Which naturally makes you wonder, if you can suddenly see the neighborhood these cancer cells are building, what does the architecture actually look like?
Well, when the researchers looked at how the tumor cells physically cluster together, what they call patches, they realized the different subtypes build completely different types of cities.
How so?
The basal-like and immunomodulatory subtypes, they tend to organize into a few massive, highly proliferative patches. They act like giant, aggressively expanding urban sprawls.
Contrast that with the luminal androgen receptor, the mesenchymal and the mesenchymal stem-like subtypes.
Right.
The map showed that they don't build megacities at all. They are scattered in many small, highly metabolic patches. They look more like isolated rural villages dotting the landscape.
But the most profound revelation of this map actually had nothing to do with the tumor cells themselves.
Wait, really?
Yeah, it was all about the stroma.
Ah, the stroma. So for context, the stroma is the supportive connective tissue that surrounds the tumor. It's the scaffolding, the blood vessels, the immune cells.
Exactly. And when the researchers used that deconvolution math to separate the tumor cells from the surrounding stroma, they found that the traditional bulk subtypes we've relied on for years were essentially lying to us.
Because those old blender subtypes weren't just describing the cancer cells, they were heavily driven by the surrounding scaffolding.
This completely upends how we view the disease. Let's take the mesenchymal stem-like subtype, or MSL.
Okay.
For years, bulk sequencing told oncologists this was a distinct, unique type of cancer cell. But the spatial map revealed that MSL is very often just a standard mesenchymal tumor that happens to be sitting in a very specific immune-rich stroma.
Whoa, so the cancer cells are identical, but the neighborhood is what changed.
Yes. The real estate around it completely flipped its classification in the blender.
The real-world stakes here for a patient sitting in a doctor's office are massive. Are you saying 2 patients could have the exact same biological tumor cells but receive a completely different subtype classification just because of the surrounding real estate?
The data proves it, and the clinical implications of that real estate are literally a matter of life and death.
Give us an example.
So, the researchers isolated a group of patients who all had identical mesenchymal tumor cells. Some of these patients had a standard mesenchymal-type stroma surrounding their tumors.
Oh, okay.
But others had an MSL-type stroma surrounding their identical tumors.
So what happened when they tracked the survival rates of those 2 groups?
The patients with the MSL stroma, which, remember, is rich in immune activation and inflammatory signaling, had significantly better distant relapse-free survival than the patients with the standard stroma.
That is mind-blowing. The tumor was exactly the same. The soil it grew in dictated the patient's survival.
Precisely. If a doctor was still using the old smoothie approach, they would have grouped some of those patients together, totally missing the fact that their survival odds were entirely different based solely on the neighborhood.
The soil matters just as much as the seed.
It really does. And understanding that the soil can actively suppress or fight the cancer brings us to the next massive question.
What exactly is hiding in that supportive stroma that acts like a localized army?
And this might be the most fascinating discovery in the entire paper. Tertiary lymphoid structures, or TLS.
TLS. Okay, to understand why these are so incredible, we have to look at how the immune system normally operates, right?
Right. Usually, if you have an infection or a cancer, your immune system launches its attack from your lymph nodes.
Lymph nodes are established, permanent military bases scattered around the body, but they are often physically distant from the actual tumor site.
But the immune system is highly adaptable. Sometimes it does something extraordinary and builds an ectopic lymphoid organ.
Ectopic simply meaning occurring in an abnormal place.
Exactly. The immune system literally constructs a temporary, highly organized fortress right next to the tumor, sometimes directly inside the stroma.
They act as forward operating bases.
That's a great way to put it.
The immune system recognizes a massive threat and decides it doesn't have the time or resources to constantly march soldiers from the distant lymph nodes.
Yeah.
So it just sets up a frontline command center right on the edge of the battlefield.
And inside these TLS fortresses, the biological coordination is stunning. You have B cells actively producing antibodies, T cells preparing to launch physical attacks on the cancer,
And specialized cells presenting tumor antigens so the local immune system knows exactly what the enemy looks like.
Exactly. But historically, we just couldn't study them. TLS are incredibly tiny and quite rare.
In a blender, the quiet genetic signal of this tiny fortress gets completely drowned out by the screening noise of the massive tumor.
Right. But with our 100 micrometer spatial map, the noise is no longer an issue. Because they kept the geography intact, the researchers could look specifically at the exact coordinates where the pathologists visually spotted these fortresses.
That's the advantage of the map.
By isolating just those specific barcodes, they captured the exact gene expression of TLS for the very first time. They basically distilled this complex fortress down to a highly specific 30-gene signature.
A 30-gene alarm system that proves a fortress has been built.
Yeah, and when we look at the function of those 30 genes, it is a perfect mix of military logistics. Some are dedicated to building B cells, several prepare the T cells for combat.
But crucially, there are priming genes, right? Specifically one called CXCL13.
Yes, CXCL13 is key.
You can think of a priming gene like a chemical flare gun. The fortress fires it up into the local tissue to recruit even more circulating immune cells to the front line.
And the clinical impact of being able to test for this 30-gene signature cannot be overstated. High levels of this TLS spatial signature are strongly associated with a much better prognosis for the patient.
That makes sense. You have a stronger army.
But even more importantly, it is highly predictive of how well a patient will respond to modern immunotherapy.
Right, because immunotherapy works by essentially taking the biological brakes off the patient's own immune system, allowing it to attack the cancer at full force.
Exactly.
But taking the brakes off only works if you actually have a car fueled up and ready to drive on the front lines. That is exactly what the TLS provides.
And the researchers proved this concept extends far beyond just breast cancer. They took this newly discovered 30-gene signature and tested it against external, completely independent data sets for different types of cancer.
Wait, they applied a breast cancer signature to other diseases?
They did. They applied it to metastatic melanoma, pancreatic cancer, and bladder cancer.
Wow. And what did they find?
Across the board, this exact 30-gene signature accurately predicted which patients would survive longer and which would respond best to immunotherapy.
So the architectural strategy of building a frontline immune fortress is universally effective.
It appears so, yes.
Yeah. So before we look at the final piece of this puzzle, let's do a quick recap. We moved from the blender to a microscopic map, discovering that the scaffolding around the tumor often dictates survival.
Right.
Hidden inside that scaffolding, we found temporary immune fortresses and extracted their 30-gene signature, proving we can predict who will respond to immunotherapy.
Which is huge.
So the final question is, what happens when we zoom all the way out and look at the entire ecosystem of the city at once.
Zooming out reveals the most transformative phase of the study. The 9 neighborhoods of TNBC, formally, these are called the spatial archetypes.
Spatial archetypes.
Okay. To build this macro view, the researchers took a bit of an analytical nesting doll approach. They started with those 270,000 individual spots. They grouped similar spots together to find 14 megaclusters.
Megaclusters. So you can think of these megaclusters like broad city zoning laws.
Exactly. A megacluster might represent a commercial zone or an industrial zone.
In biological terms, one megacluster might be heavily proliferative tumor cells mixed with dense stroma, while another might be mostly fat tissue and blood vessels.
Right. They then looked at how these 14 megaclusters mix together within individual patients.
I see.
So if a patient's tumor ecosystem is 50% commercial zone and 50% residential, they represent one type of overarching city.
And if another patient is 80% industrial and 20% parks, they're a completely different biological landscape.
Yes. By categorizing these diverse landscapes, they discovered 9 completely new spatial archetypes, or SAs, for triple-negative breast cancer.
9 distinct landscapes.
This is the new map of the disease. It completely supersedes the old 5 bulk subtypes because it accounts for the whole ecosystem, not just the tumor cells.
So analyzing these 9 spatial archetypes must reveal drastically different patient destinies.
Night and day differences? Let's look at spatial archetype 4, or SA4. This is the archetype a patient desperately wants to see.
Why is that?
It is highly immune infiltrated and has extremely high levels of that TLS30 gene signature.
The immune fortresses are fully established and fighting.
Exactly. The survival data for SA4 is incredibly promising. Patients with this archetype had the best survival rates of the entire 94-patient cohort.
That's amazing.
The prognosis is so overwhelmingly positive that researchers are suggesting these patients might actually be candidates for de-escalated treatments.
Meaning they might not need to endure the harshest, most toxic chemotherapy regimens because their own immune fortresses are already doing the heavy lifting.
Right. That is a massive potential win for a patient's quality of life.
But on the opposite end of the spectrum, you have spatial archetype 8, or SA8.
Yeah, this is the dark side of the coin. SA8 has almost zero immune activity. The tumor has engineered a cold, barren landscape where the immune system simply cannot establish a foothold.
Unsurprisingly, patients with the SA8 archetype had the absolute worst survival rates.
It's a very tough landscape. But the archetype that really stood out as a paradigm shift is SA5.
SA5. SA5 is exclusively made up of tumors from the luminal androgen receptor bulk subtype.
Right, but looking at its spatial geography, it is a completely isolated, unique metabolic landscape.
Unlike all the other archetypes, SA5 is heavily driven by lipid metabolism, meaning the way the cells process fats for energy.
Exactly. And when you see an ecosystem that's just different tumor structures, different stroma, a completely different metabolic engine, it begs a pretty major question.
Does it even make sense to keep calling this triple-negative breast cancer?
And the researchers share your skepticism. They formally proposed that the luminal androgen receptor tumors, specifically this SA5 archetype, should potentially be reclassified altogether.
Wow. So lumping it into the TNBC bucket is actually doing a disservice to the patient.
Totally. It behaves so radically differently from the other archetypes that it should likely be considered an entirely distinct entity of breast cancer, standing on its own alongside standard luminal, HER2 positive, and TNBC.
Which means the way an oncologist approaches treating it would have to change entirely. You wouldn't use standard blanket chemotherapy.
No, because SA5 is driven by the androgen receptor and highly specific lipid metabolic pathways. Hitting it with standard chemotherapy is like trying to put out an electrical fire with water.
It's the wrong tool for the job.
Exactly. The researchers suggest SA5 requires targeted, unique therapies. Specifically, they point to anti-androgens combined with PIK3CA inhibitors.
Okay, a PIK3CA inhibitor. For you listening, that is a highly targeted drug designed to throw a wrench into a very specific mutant enzyme that drives this unique metabolism.
Right, it shuts down the specific engine the SA5 tumor relies on.
Looking at the disease through this spatial lens really shifts the entire landscape of oncology. I mean, we have successfully moved from blending tumors into a chaotic smoothie with bulk RNA sequencing to meticulously mapping them on a 100 micrometer grid with spatial transcriptomics.
It's a huge leap forward.
We uncovered the secret life of the stroma, realizing the soil dictates survival. We found the hidden immune fortresses, extracted their signature, and proved they predict immunotherapy success across multiple cancers.
Yeah, that 30-gene signature is a game changer.
And ultimately, we mapped the 9 spatial archetypes that could completely revolutionize precision medicine, ensuring the right patient gets the right drug at the right time.
It is a profound reminder that in biology, context is everything. The individual cells matter, sure, but the neighborhood they build matters just as much, if not more.
It changes everything about how we look at disease. And I want to leave you listening with a final thought to mull over. We have just explored how simply mapping the geography of cells completely rewrote our understanding of a complex, deadly disease like breast cancer. So, what other massive mysteries in the human body are hiding in plain sight? From the tangled amyloid plaques of Alzheimer's in the brain to the localized misfiring of cells in autoimmune diseases. How many medical breakthroughs are just waiting to be discovered the moment we finally stop looking at the smoothie and start looking at the map?